now, I tend to record all of my lectures for you. So if you want to record on your own, there's something in the syllabus about you have to get my written permission. But because I project from a tablet that can also record everything that I'm doing, both using my finger on the screen and also audio, at the end of every class then, when I get back to my office or when I get home at night, I will upload these movies from class. The movie is just what you see on the screen. It's not video, it's the screen plus audio for your use in studying. If you miss a class and you need to review what happened in class, those will all be on YouTube. You can go to my channel on YouTube, and tonight there will be a Biology 102 Fall 19 channel that I've established with today's recording. So one beautiful thing about Discovery classes is my using a tablet means I get to provide extra resources for you. So a little bit of introduction, then we'll get into what the course is all about. So like I said, I'm Dr. Ross. This is me in my lab in Science 1, staring through a microscope at the worms that I study. The maybe most important thing about this is, depending on my hairstyle at the time, you'll see I've got a giant scar on my head. That wasn't a fishing accident or Jaws or any weird shark bite or anything. I actually had brain surgery a couple decades ago. I'm good, but that's just the icebreaker. So if you see the giant scar on my head, it's all good. Don't talk about it anymore. Although if you do want more information, I'm happy to give it. But more than that, what I did in grad school, which was actually I was halfway through, I was in my third year of grad school trying to get my PhD when I found out I needed to have brain surgery. So I took like a half a year off to have brain surgery. Thank goodness for insurance too, because like a couple $100,000 of medical bills is not fun during school. Eek. But what I was working on both sides of having brain surgery, was I was studying chromosomes in this particular species of fish, the stickleback fish. This is actually a 3-spine stickleback fish. This is a male on the bottom right. And we can tell it's a male because males, when they're ready to mate, turn bright red on their throat and they get kind of bluish back, compared to a female, which is silvery pretty much her whole life, regardless of whether or not she's ready to mate or not. So this is a sexual cue that males develop when they're ready to mate, and then they show off to the females and try to attract the females to their nest. But I was not interested. Everybody else in the research lab I was in was studying the behavior of these fish. And I was interested in studying their chromosomes, and hence genetics. One of the things that I learned, and you don't need to know this is not on the test, it's just an introduction of me briefly, is that like humans, these fish, these 3-spine sticklebacks, have an X chromosome and a Y chromosome. So what do Y chromosomes do in humans? Anybody? Determine gender. Yeah, sorry, they determine sex. Big difference. Y chromosomes make individuals male in humans and in this species of fish. The weird thing is human Y chromosomes are these tiny shriveled up little chromosomes that have lost most of their DNA, and they basically all they do in humans is determine whether or not you're male. And what I discovered in this species of fish was I used these brightly colored dots here, aside from the laser pointer. The green and the purple are fluorescently labeled pieces of DNA that I designed to show me different genes on the X chromosome and the Y chromosome. And what I discovered was that the X and the Y chromosomes are differently shaped. You can, it's not easy to tell in any of those images. But it turns out that the 3-spine stickleback's Y chromosome hasn't fallen apart, literally fallen apart, DNA just falling off, as much as the human Y chromosome has, which is interesting. So it's only interesting because the Y chromosome slowly degrades over time. DNA basically jettisons itself from the chromosome. And so I'm interested, or was interested in understanding how that process worked physically. So how does chromosome break? How does a chromosome break? How does DNA leave a chromosome? How do chromosome sizes change? Then I mentioned that I currently work on worms. So as a postdoctoral researcher, after I got my PhD, and then when I was hired here in 2012, since then I've been working on worms. Little microscopic worms, not earthworms. They're about 1 millimeter long. It's about the size of a 12-point font comma when they're adults. So they're microscopic. And this is a, not of course to scale, image of one of the worms. Their genus and species is Caenorhabditis briggsii. Has anybody heard of C. elegans? A few of you have heard of C. elegans. Common model organism for genetics. This is a sister species. It's the same genus, very closely related to C. elegans. But what's really interesting about these species is that there are very genetically diverse populations of worms. So people that are scientists, biologists, ecologists, have gone all over the world collecting this species of worm. And in this case, there's this population that is called by the community AF16. It's from Japan. And there's another population, sorry, HQ104 is from Japan, AF16 is from India. Right? Tropical and temperate regions. And if you mate those individuals together, if you take an Indian and a Japanese C. briggsii and you mate them together, their offspring aren't quite as healthy as a just Indian worm or a just Japanese worm. And this is very similar to horses and donkeys. What happens if you mate a horse and a donkey?
Mule.
You get a mule. The mule is sterile. So this is a genetic issue. In horses and donkeys, we know what the issue is. Why, when they mate, when the sperm from a horse and the egg from a donkey fuse and you make a mule, the reason the mule's not healthy is because the horses and the donkeys have different numbers of chromosomes. When those different numbers of chromosomes combine in the offspring, then meiosis and mitosis don't work properly. We're going to talk more in the semester about meiosis and mitosis. And so basically what we do in my lab then is we mate horses and donkeys, except instead of actual horses and donkeys, we use worms instead. The goal is the same though, which is to understand what are the genetic differences between different individuals of the same species, like any two of you in the room, that cause their offspring to be less healthy than the parents. Humans don't really show this yet. We have lots of different populations of humans. There are humans that live in Japan and India. But for all intents and purposes, if somebody from Japan and somebody from India have a kid, the kid's usually fine. There's no genetic differences between Indian and Japanese humans that I know of that cause all their offspring to be less healthy. This is the case in horses and donkeys and in the worms that I study. So the students in my lab now are working on trying to figure out what are the genetic differences between AF16 and HK104. What are the mutations that distinguish those two groups that when those mutations are combined in the offspring that does make their kids less healthy? Any questions about that? Again, it's just introduction. This is not on the exam. No stress. How's the lighting in the room? Everybody okay with the lighting so far? Good. No one's falling asleep because I turned off the bank of lights? Okay. So, a couple of thoughts. One is, and I mean no offense to those of you that are sitting in the front where I just turned off the lights, but when I was in your seats, when I was an undergrad, University of Oregon, I was a duck. When I was a, yeah, is that good or bad? Oh, awesome, from Eugene, fellow Oregonian. So I'm from Albany, actually, originally. Willamette Valley. So it's another valley, another agricultural area, just not as hot. And I'd always sit up in like the second row because I was that kid. And it was really embarrassing. I met my wife at University of Oregon. She was a psychology major. Actually, true story, the first time I met her, I said, "Oh, you're a psychology major? I'll never be able to marry you." Literally, I said that the first time I met her, but it turned out then 4 years later we got married. And since had 2 kids, by the way, who were both very healthy. No worries about my genetics. But so second year, after I had met my now wife, we were in organic chemistry class, and my friend and I, Bryce and I, were sitting in the second row right there on the edge, and my wife and her friends were sitting in the back, my girlfriend at that point. And somebody sitting next to them said, "Hey, do you see those 2 guys up in the front? They're the ones that always break the curve." So, yeah, we were always the ones who were up there raising our hands and answering questions freely, and I don't mind that, but what I do now ask, because I realize that that is an issue that sometimes I was the cause of, that when I ask for questions, that what I like to do most of the time is I will ask for people to raise their hands and not answer, and I'll pick on people that have raised their hands to answer, just so we get a good distribution from the back of the room to the left side of the room, your right side of the room, so that everybody gets a chance to think about an answer before responding. Is that good with everybody? Sometimes I'll just ask for brainstorming type questions. I'm going to do that in just a second. Oh, in fact, right now. So to introduce genetics a little bit more, you've all had presumably some biology classes, maybe 1A and 1B. Some of you are not biology majors. Most of you are. We've got some biochem majors in the classroom. We've got a couple of special majors in the classroom, meaning they've designed their own coursework to get a degree. So I want to make sure that we have a chance to get on the same page about genetics. So what is the first thing that comes to mind when I say genetics? So this is not a raise your hand situation. This is a, what do you think of when I say genetics?
DNA.
Okay, so DNA, classic response. DNA is the molecule that carries the hereditary information. That's what genetics is all about. It's all about DNA, and that's why we start the class with DNA. What other things do you think of when I say genetics? Protein coding. So genes, for the most part, although not entirely, encode proteins, and those are the enzymes and proteins that both make us up and help our cells run, operate. Are there any negative connotations? Those are just sort of science words.
Like
What feelings? Okay, so diseases. There are a lot of genetic diseases, but there are a lot of diseases that are not genetic. So if those of you that are pre-vet, pre-opt, pre-dental, pre-med, pre-pharmacy, we will be talking about some mutations that cause diseases, but there are other diseases, of course, that have no component that's genetic, or very little that's genetic. What else?
Family or hereditary tree.
Okay, so pedigrees
There we go.
and inheritance. So I was tinkering with showing one of my family, my handwriting's pretty bad on a tablet, but that's why you get the audio along with it. So yeah, I was thinking about showing a picture of my family and having you guess like who I'm most closely related to in the picture, because you can kind of tell that I look like my dad, same hair pattern, same ear shapes and so forth, but you probably, I hope, all have that same sort of experience, or at least can identify with the idea that sometimes kids look more like one parent than the other, and yeah, why is that? Other thoughts? Anything else? Genetics.
Dominant and recessive dominance.
Okay, so dominance, going along with inheritance. And this is often a major concern in genetics class, the concept of dominance and recessiveness, and we're going to spend a bit of time talking about those, because I want to make sure that everybody gets a chance to be clear, if they want to, about all of the topics and terminology we're going to use in the class. So that's a good approach. Good start. So, quick quiz. Think for half a minute. Here's a eukaryotic cell with a bunch of organelles labeled. Where's the DNA located? Don't answer yet. Where can you find DNA in a eukaryotic cell? Okay. Any thoughts? What do you think? Where do you find the DNA? Yes?
In the nucleus.
Okay, so we've got DNA in the nucleus. Anywhere else? Mitochondria. And if this was a plant cell, where else would you find DNA? That's okay. My dad's a botanist, actually. I'm not. So, much to his shame, perhaps. I don't know. Chloroplast also has DNA. But in an animal cell, which is mostly what we're going to focus on this term again, because I'm not really a botanist, we're going to talk a lot about animal cells and eukaryotic cells, the nucleus, but the mitochondria, what does the mitochondria do? Powerhouse of the cell. It generates ATP, the currency of energy that all of our cells need to survive. The mitochondria has its own DNA, separate from the nuclear genome. So sometimes this semester we're going to mostly talk about the chromosomes in the nucleus that are the things that you're probably used to seeing that look like X's when they're in metaphase. The mitochondria, though, have a circular genome. And the reason out is because one of the things that we do in my laboratory as good transmission geneticists, studying the inheritance of traits from one generation to the next, we cross males. So I'm going to use a solid line to represent one chromosome. These are diploid organisms, which we're going to talk more about ploidy later. They're diploid. So in the nucleus, every chromosome, there's 2 copies. So we could take one of those, let's say red, let's see that's the Indian C. briggsii, AF16. And we mate him with a hermaphrodite. That's the other cool thing about C. briggsii and C. elegans. There are no females. The females are actually hermaphrodites. They make their own sperm and their own eggs, and they self-fertilize, so they don't really need males. Males in this species are kind of like, who needs them? So if we cross in the P0 generation, the parental generation, P, we cross those individuals, then we make an F1. And the F1 generation inherits one chromosome from dad and what?
From the other parent.
It's got to be diploid still. It has to have 2 copies of every chromosome, so it gets one of the blue chromosomes as well. There's a weird thing that we'll talk about again in class this semester that's called recombination, which is that when you have an organism that's making sperm or eggs, crossovers occur. Hopefully your eyes aren't glazing over from high school biology. Oh, Jesus, crossovers. It'll be easy. You get recombination between the chromosomes, and that means that in the next generation you can get part of one chromosome that's red stuck onto the rest of the same part of the same chromosome, but that's from the other parent. And that's how, presumably, we get all the variation that I can see out here in class. It's because we're not all identical to each other because of recombination. All of our chromosomes differ a little bit because of recombination. So in this case, a single organism would have another chromosome, a second copy of the chromosome that's also recombinant. So that's one of the things we do in my research lab is we mate the Indian and the Japanese populations of C. briggsii, and we get F1s. And because these are hermaphrodites, part male, part female, we can just self them. So we take a single F1 individual, let her, it, whatever, produce its own offspring, and that's the F2 generation. And every then brother or sister in this F2 generation is a little bit genetically different. They're all siblings, but they all have slightly different chromosomes because of recombination. So let's make a prediction. I'm going to ask you to make a prediction. Are all of those F2, the grandkids basically, are all of those siblings going to be equally healthy?
No.
If I hear no, not necessarily. So what would cause those siblings, brothers and sisters in that grandchildren generation, what would cause them to be different?
The different recombination of the chromosomes, different crossing over, having different parts of different chromosomes.
Right. Exactly, which unfortunately I just said, so I gave the punchline away before I asked the question. But you're absolutely right, so it's a good reiteration. The siblings are brothers and sisters of each other, but they're genetically different. And one of the things that we learned, and this is going to take a while to load because it's a big image file. One of the things we recently learned in my lab, it's studying the genetics then of health in worms instead of humans, is that if we measure different siblings across time, so here time runs across the top of the screen. So time in hours. So we photograph worms at 24 hours of age, 36, 48 hours, and so forth. And these worms they take about 3 days to become adults. So at 72 hours, you probably can maybe slightly see, let's pick this one for example. That's one photograph of one worm. The worm is the dark squiggly line, okay? Adult worm. So for those of you in the back, hopefully you're looking at the slides on your own device as you can, because you can always download the slides, PowerPoint, or PDF file rather, from Canvas before class. I always upload them beforehand. So if you don't have a good seat in the house, if you don't want a good seat in the house, you can always follow along on your device and take notes on the slides too if you want to. So at 72 hours, all the worms look like they are actually the same size. We measure them digitally, and they're all the same length. So we're using length to represent health. How quickly, in other words, worms grow. From 24 hours to 72 hours is what we're measuring. And we find that some of those F2 individuals are basically adults by 52 hours. So that's about the time when worms normally reach adulthood. But some of their siblings, can you see those 3 worms? Those are 3 different wild type worms. Those are normal, this is normal growth, up to 52 hours. Can you see how big those worms are? Is it easy to see? They're kind of the dark squiggles in there. Compare them to the next 3. So these are siblings of theirs. So here at 52 hours, what's the relative size of those three brothers and sisters and those three? Who's bigger?
The wild type is larger.
Right, the three wild type worms, these are just representative siblings, are smaller, or bigger than the F2s here, the grandkids that are their sisters and brothers, but they don't develop as quickly, they grow slow. So right now, one of the projects in my lab is trying to figure out why, even though those siblings, all of these F2s have the same grandparents, what genetically do these that are delayed in development, that grow slowly, have in common with each other? What are the genes that cause some individuals to grow slowly? My youngest daughter actually is a slow grower too. She's not developmentally delayed, but she's the smallest kid in her class. And so I kind of wonder if there's a genetic basis of that too. One of the nice things about this class is we can skip content if we want to, and since I've taken a little bit longer on that, then we're going to skip the second example of a type of project we do in my lab. So wrapping it all up then, there are 4 big concepts we're going to talk about in class this semester. 4 different sort of branches of genetics. And this is the way that we're going to cover these concepts in class, and the way that the course manual has been developed is around these 4 topics. Molecular genetics, which is what we start with, is all about the structure of DNA. We'll be talking about nucleotides, chromosomes, chromatids, those sorts of concepts. So we're going to start with what is DNA. That's not usually, do you remember the last time you learned about genetics? What's the first thing you learned about? How does it normally get introduced?
Chromosomes.
So sometimes it's chromosomes. Who's the most famous geneticist you know? Mendel. So a lot of textbooks start with, this is Gregor Mendel, 1600s, studying peas, crossing them together, wrinkly peas, yellow peas. Right. I feel like we should learn about what DNA is. Even though Mendel didn't know about DNA at the time, we should know about what DNA is, and then we'll get to, eventually, transmission genetics, which is inheritance, pedigrees, traits, how diseases pass from generation to generation. And in between those two, after we learn about DNA, we're going to learn about the central dogma. That's all about gene to messenger RNA to protein, how DNA encodes the proteins that make us up and help us survive. And then the last topic, the third topic we're going to talk about is gene regulation. Did you ever realize, maybe you did, maybe somebody told you, maybe it hasn't been a while since you thought about this, that every cell in your body has the same DNA? It's kind of I don't know, if you know about DNA or if you've taken biology before, you might have already realized this, that you each started from a single cell, and now every cell in your body has the same DNA. So the question that a lot of geneticists are interested in terms of gene regulation is, how do I have skin cells and fingernails and bone cells and blood cells, because they all have the same instructions inside them. They all have the same DNA. So how does an eye cell know that it should become an eye cell and be able to build proteins that absorb light and help me see, as opposed to being a muscle cell, which basically just contracts and expands and contracts and expands? So that's all in gene regulation, is how does a cell know which genes to turn on and which genes to turn off so that we can be complex organisms that still mostly function. Okay, so that's a brief intro to how we're going to go through the topics this semester, a little bit about what genetics is going to be about. So now we get down into a little bit more of the nitty-gritty details of how the class is going to run. So today I'm going to have you do a quick syllabus quiz. It's just graded for participation. I just want to know what you remember from the syllabus. So those of you that didn't look at it yet, don't stress out. It's just for participation. There are 2 main computational tools that I'm going to use during class and outside of class to get your feedback and to collect assignments from you. One of them is called Socrative. It's a polling or quizzing app that I asked everybody to install. If you read the syllabus, you knew to install it. But if not, you've got time to do that. The other apps we're going to use a lot are Google apps, including Google Classroom. So you should have that app installed on whatever device it is you're going to use. If you've got a laptop, it's not an app, you can just use it through a web browser. So the first few days of class, it's going to be part content and part practicing using these tools. Because I don't want to get to the first exam and nobody have tried using Google Classroom before, and then everybody's stressed out about, does the technology work, not just do you know the material on the exam. So we're going to go a little bit slowly these first few days in introducing some of these tools and practicing using them. Everything we're going to do is worth points, but small amounts of points and for participation for completion, not necessarily for the quality of what you turn in. Okay. So the first thing we're going to do today is a syllabus quiz, and then later today I'm going to talk about a few surveys that I've prepared, not all of them to do today. And I just want you to know from this slide that if you go to Canvas and go to assignments, I'm always going to put the in-class assigned assignments here, but we'll work on them in class. So if you see something pop up that says it's a survey or it's Socrative, don't do it outside of class, because those are things that I'm going to prompt you to start working on in class. So with that said, here's the Socrative quiz. So you can do one of two things right now. You can either open a web browser and go to Socrative.com. That's it. Just type in Socrative.com, go to that website, and you'll want to pick, you'll be asked to log in as a student or a teacher, so I'll let you decide which one you think is appropriate. This is your first test. If you choose student login, then you'll enter room name Ross Genetics, all one word. And that's how you get from the big giant website Socrative to the quiz that I've got for this class today. So you don't have to enter your information, just that in the room name field. If you've got an app, the Socrative student app, you do the same thing. You open the Socrative student app, it asks you for the room name, you put in Ross Genetics. Are there any concerns or questions at this point? We do have one of our Discovery staff, our guide on the go in the back. So if anybody wants in-class tech help, we've got an additional resource.
I'm waitlisted in the class. Can I still?
Yes, if you're waitlisted, you can definitely get in and do this. Absolutely. Everybody who's in the room, if you've got a device, you can do this. Smartphone, tablet, it's all done. So you hopefully see a screen that looks like you're waiting for something to happen. If you're not seeing something like that, please let us know. So let me show you what I'm doing on my computer, just for full transparency, because I will never lie to you, and if I am, I'll tell you that I'm lying to you. I want you to see what's going on. So I'm about to start this quiz, and as soon as I start the quiz, then you should all get pushed to the first question in this quiz. Again, just for participation points. And so you can see what I've done is I've turned off student names. It's asking you to give your student name in the quiz. That's because I want to know who is here, because this is attendance for the day. But up here, I can see your names popping up. Right? First question, second question. Now I have to turn this off because you're not supposed to see each other's student ID numbers. But the point from that is that I see in real time your responses. And the reason that I like using Socrative to do this is because at the start of most classes, I'll give a brief quiz, kind of like this, 2 or 3 questions max. I want to figure out how much people know and how much people don't know, what I should focus on talking about that class period. So I'm very dynamic about what we do in class, and this is one of the ways that I'll get that information from you. So if you're worried about whether or not you're getting the right answer, this time and this time only, you won't know which of these people you are, but I'll show you what is going on in class. We can start to see trends emerge. And then we're going to briefly talk about the syllabus, and we'll move on to a couple more things before we end. Bless you. I didn't do a very good job of randomizing the true and the false, apparently. It's either tricky or it's not tricky, I'm not sure. So like I'm doing now, at the start of every class, as your answers roll in, I'm going to see if it looks like there are any confused patterns, right? The first 5 questions, it looks like everybody knows more or less what's going on. But maybe these last, like this question, true, false, we've got 63% right answer. So, finish that up. When you're done, you just finish the last question when you submit it. There's no submit button at the end. As soon as you're done answering the last question, what does it say? Yeah, waiting for another activity. So when you see that screen, you can close. You don't have to do anything else to submit. Okay. So what was that second to last question? Letter grading is on a 90, 80, 70, 60, 50 ABCDF scheme. That was that question. This one that is about half of you are saying true and half false. That's false. So this minor issue, minor point on the syllabus, I don't expect any of you would have necessarily recalled that, but I want to make a point about that. And I'm going to go over a couple other really quick points about the syllabus as some of you finish up. So, discovery class, really quickly. Discovery class, please bring your devices fully charged. You can tell, as most classrooms on campus, there are not a lot of wall outlets. And I do want to make sure you can participate, so please bring your device charged. The way class works is every week, or every class period rather, I will have a list of a couple of chapters from our free OpenStax textbooks. They're PDFs you can download. Has anybody bought one from the bookstore? I'm not sure if the bookstore did any print copies. Sometimes the bookstore makes some print copies for those of you that would rather have print than PDF. I'm seeing heads shake.
For the OpenStax?
For this class's OpenStax, there are 2 OpenStax textbooks in the syllabus that I'd like you to download, please. They're both PDFs, they're both free. So that information's in the syllabus. The other course content we're going to use, there are 2 other types. One is the course manual that I'm writing, and I've uploaded that to Canvas, so you can access, whenever we get to each particular content, I'll tell you the day before, please read this chapter of the manual for next class. Those are free, those are PDFs, those are on Canvas also. In the course manual, the textbook that I've written, there are links to videos that I've pre-recorded on certain topics that I'd like you all to watch in advance of class. This is a flipped class. It's sort of my approach to teaching. It's also called blended learning. It's you prepare before class, we spend in-class time doing. It's a little bit more active learning, so it's not you sitting there, me talking like I have been today. It's more you come to class having watched the videos, read a couple of sections of a couple of chapters, so I'm not asking you to read whole chapters of anything at any one time. I'm trying to keep the coursework lighter or reasonable. But you prepare in advance. The way you get something out of the class is if you're prepared to talk about what you learned or what you didn't learn in class. This is the most expensive part of your tuition dollars, is you're paying for not only the buildings, but for my time. And I feel like I can make my time most useful to you if in class, when we're all here, I can help you with whatever concerns or problems you're having with the content. This is going to be a little bit uncomfortable for those of you that expect these large-ish lecture classes to be lecture. This is not going to be always lecture. It's more going to be in-class activities, discussion, question and answer. I love it when I come to class and all I do is answer questions. As long as I can format it in a way and help everybody bring each other up to speed so that I'm not wasting anybody's time. But usually, as you know, if one person has a question, then half the class has a similar question. Okay. So all of those course materials, the links to videos I'd like you to watch, the chapters, sections of chapters of OpenStax textbooks I want you to read before class are all in the class manual. All of that's available on Canvas already. Any questions about that? So this is not the type of class where you can get as much out of it if you don't prepare in advance, which I know is a difficult thing for me to ask. I was not that student when I was an undergraduate. I never read the textbook before I went to class. I just went to class, I wrote down what the teacher said, that was it. This is not designed to be that style of class. So grading-wise, it's not 90, 80, 70, 60, 50, it's 80, 60, 40, 20, 0. So if you get 80 to 100%, it's an A. So everything's spread out evenly, basically. Bottom 20% of scores, not of the class, there's no curve. If you get in the bottom 20% of all points available, you get an F. The next 20%, 20 to 40% of the points is a D. Right. So I like to spread out the curve a little, the grades a little bit more. That gives me finer ability to assess A's, B's, C's, and D's, as opposed to crunching them all up in the top half of the number of points. For that reason, it's worth noting that there are 4 tests, 3 exams and a final. First exam is worth 10% of the points in class, so that's half a letter grade. 20% of the points is a letter grade, so first exam is 10%. The second and third exams are 15%, so it's almost a letter grade worth of points, but I want you to be able to see what my exams are like before you have to, sacrifice a letter grade's worth of points on one exam. I don't think that's really fair. And then the final's worth 5%, a quarter of a letter grade. A couple other things, but not much. Oh, I already told you that. Yeah. If you're absent in the future, let me know. Back to the tablet for the rest of the class. Okay, so if you go on Canvas, everything is going to be in the Files folder on Canvas. It's the first semester I've used Canvas. I'm still learning how to use Canvas. So instead of making modules and pages and so forth, basically I'm putting the files in the Files folder. This is where you're going to find any background literature that is in the course manual that I ask you to look at. And there's the course manual files themselves, all in the Files folder on Canvas. So for example, here are a couple of the course manual files, just a screenshot. For next class, what I'm going to ask you in a couple of minutes to do for next class is to read the cover, which is just the cover of the manual. So good luck. It's not very exciting. And the introduction to our first section, molecular genetics, which is DNA structure. So each of these 3 is like a page or 2 long. And DNA structure PDF is the actual content for next class, talking about chromosome structure, nucleotides, and those sorts of things. Okay. So at the start of class, this is the last thing that we're going to work on briefly in class today, and then there will be a few assignments, surveys I'd like you to take before the start of class on Thursday. There's a little bit to do between now and, Friday, and you'll get it done. So if you're on Google Classroom, and if you're not right now, that's okay. This is due by the start of class on Friday. But I wanted to give us a little bit of time. We've got 5 minutes or so to work on getting into Google Classroom. And there's an assignment posted when you log into Google Classroom. It's called Informed Consent Form. And the purpose of this is, because I'm a scientist and I like to know if what I'm doing is helping you, I actually am the primary investigator of a educational research project on campus that's trying to understand if students that work with mobile devices in classes do better academically than students that don't use tablets, smartphones, laptops in classes. And as the primary investigator, the leader of that research project, I would like all of you, because you're in a a discovery class, to be part of that program, part of that study. So this informed consent form describes, it's just a single page PDF form, but it's a form. I would like you to read please, and decide whether or not you want to participate in the study that I'm conducting. The only thing that really is happening is I'm going to teach the class like I normally teach the class. Whether or not you opt in or opt out of participating is you will do the exact same thing in class regardless of whether or not you choose to participate. The only difference is whether or not I can analyze your data, that is your assignments, your exams and so forth, to do this study after the semester is ended. So what's going to happen is, like I said, all the exams, you'll take the same exams regardless of whether you participate, we'll do all the same assignments. At the end of the semester, if you participate, I anonymize, so nobody's name or student ID number is associated with anything. And then I study from the start of the semester to the end of the semester, did the average anonymous student improve or stay the same, or what's the opposite of improve? Deprove? Decrease, not deprove. So there's very little risk to you, but it's totally up to you whether or not you decide you want your data from class being compiled with everybody else's class data to help us, Fresno State that is, understand if this type of class is helpful or not. So that's the whole idea behind this assignment, but the real reason that it's worth doing is because this is the first time we've used Google Classroom. And I want to make sure everybody's comfortable with getting a PDF file from a Google Classroom assignment and then drawing on the PDF file with your device, editing it, annotating it, putting your signature on that electronic file on your device, and sending it back to me. Because this is how we're going to take exams in the class. No paper. Everything's digital. So on exam days, I will put a PDF of the exam on Google Classroom. At 1 o'clock, you will download it, you'll open the assignment, you'll open the PDF on your device, you'll fill it in. You'll send it back to me electronically by the end of the class period. So yeah, I said you've got an open internet exam, basically. Because I'm asking you to do your thing on your computer. I'm not going to come around and see if you're looking at Wikipedia articles or anything. Just a couple minutes, thanks. And so that's how we're going to do exams, and because I want you to be able to practice doing these processes before we get to exams, that's one of the reasons that we're doing this. So even if you don't want to participate, I still need you to do this assignment. It's worth completion points. I need to see that each of you on your device can download a PDF, sign it, or do something with your finger that writes something on this PDF and send it back to me. You've got until Friday at the start of class to do this. So now's a good time to start. I'm going to end before you start working on that. Just got a couple minutes left. Next class, we'll also have our guide to go in class to help with any in-class technical issues if you're having problems with these sorts of things. But between now and Friday, please go to the Discovery Hub, ground floor of the library, if you wind up having any issues with doing this process.
There isn't anything posted.
Pardon?
There isn't anything posted.
Well, let's see. Well, so we'll troubleshoot in a bit. Now, one last thing to say. There are a couple of surveys that I'd also like you to, these are short, a couple of surveys that I want you to take before the start of next class, please. They're due, just participation points, 1 point each, at the start of class. These URLs are also already on Canvas. These are the two of the assignments that have already been posted on Canvas, so you don't need to write these down if you don't want to. You can go to Canvas and get these URLs. Finally, so there's the summary. And again, we've got 1 minute left. There is another class that comes in after our class, Mondays and Wednesdays. Fridays after class, we're a little bit more relaxed getting out of here. But on Mondays and Wednesdays, I also teach immediately after this class, which means I have to go right now to my next class, so I can't hang around and answer questions Mondays and Wednesdays. Fridays, sometimes I'll be able to hang around after class. So next class, these instructions are posted in the PDF of these slides that are on Canvas. So when you need to go back and figure out what you're supposed to do for Friday, this is the list, the course manual PDFs that I already mentioned, the 2 surveys, this Google Classroom assignment, just sign your name and send it back to me. And the last request, which is going to make everything way smoother, if everybody is on the EduRoam Wi-Fi network, not Bulldogs, not Fresno State. Please go to this URL if you're not already on the EduRoam Wi-Fi network and get connected there. Again, you can ask the Discovery Hub for help on that. That's not necessarily, I'd ideally like that to be done Friday, but definitely before the first exam. Things will be way better if we're all on that network. Okay. Thank you for your participation today, and I will see you next time.